everybody. Welcome to a mini lesson on pedigrees. So this is some information from chapter 16. We're not covering all of chapter 16, just a few of the major topics that apply to human genetics. So pedigrees are a tool that geneticists use and historians use to try to trace or track a trait through many generations. So in chapter 11, you learned how to use a Punnett square, which, if you remember, a Punnett square was a tool that allowed you to predict the genetic combination of offspring. So if you had a heterozygous parent crossing with a homozygous recessive offspring, you could fill out your Punnett square, right? And that said, it gave you information about what's the percentage. If a heterozygote crossed with a homozygous recessive, you would have 50% heterozygous and 50% homozygous recessive. So that's kind of predicting the future. Pedigrees allow you to not only predict the future based on matings and crossings, but it also allows you to look at the past, to allow that information to help you determine maybe there's some mysteries or some question marks in the familial history that you don't have any data or records for. So pedigrees help you figure that out. So there's some symbols we're going to take a look at, and then we'll look at a couple examples so you can be comfortable learning how to solve a pedigree, so to speak. So pedigrees are charts, and you'll see one in the textbook, and you'll kind of see one on these examples. But the way that they're written is they're a series of shapes, usually boxes and circles, and lines indicating relationships. So if you have a square is a male, and a circle is a female. If you did see a diamond, it just means the sex is unknown. So maybe it was an infant and it died before any of the records were kept or something along those lines. If you are a empty shaded shape, so like a empty square or an empty circle or an empty diamond, for whatever trait that you are tracking, you are unaffected, which means you do not have that characteristic. If you are shaded, if the box is shaded, it means you have that trait. So they're just saying unaffected or affected. It just means if you're tracking freckles, if you had freckles, your shape would be shaded. If you didn't have freckles on your face, your shape would be empty. If you had a widow's peak, right, of the hairline or a straight hairline, whatever the trait that you're tracking, if you have it, you would be shaded. If you wouldn't, you'd be empty, empty box or an empty circle. When you have two shapes that are aligned or attached by a single line, this would be a mating, typically known as a marriage, but that always doesn't have to be the case. And then a vertical line is showing, so let me highlight these, right? So horizontal line is a mating. So here's a mating between a male and a female, and then a vertical line is an offspring. So that's a son in this particular case. For a family, you would have your mating, you'd have your offspring, and then another horizontal line is showing you the family. So all of these would be siblings. So we have a brother, a sister, and non-identical twins. If they were identical twins, they'd have to be of the same sex, so both boys or both girls, and usually there's a little line, a horizontal line connecting those two branching bars. Now down here, I didn't mention this double line. This is what's called a consanguineous marriage. You want to take a guess? I usually ask that question in class. Consanguineous means incestuous or mating between two closely related individuals. And that's not always a good thing because it increases the chance of recessive traits being expressed in the offspring. And that's not always good. So this is the key. So there's a key in your textbook. You can reference that, or you can come back and look at this video or the PowerPoint. So squares are males, circles are females. Shaded box means you have the trait. An unshaded box means you don't have the trait. And then your various lines are showing you the generations. Now, what we can do, kind of linking it with information that we have learned in chapter 11, this first mating, this could be your parental cross. And then these offsprings could be your F1 generation. So kind of think about what's happening. So if this was our big T, and little t, that would be what we would set up like I had in the Punnett square on the first slide. those would be the potential outcomes. But then here would be the actual data where Punnett square gives you percentages, a 50-50 chance on a dominant phenotype versus a recessive phenotype. You would actually get that information. So if this was the case, I would fill this box in, right? Because he's dominant. If this is a dominant trait, he would have that dominant trait. And then there could be, maybe the son and one of the twins has it. So that lets you know, it's kind of a predicting, based on your Punnett square, that this particular individual has to be big T, little t, because that's the only way that those, the law of segregation, that's the only way that they could have, the genotype that they could have that phenotype. If they're unaffected, it has to be homozygous recessive, and that would have to be the phenotype for those particular individuals because their boxes are unshaded, and that's the only combination that they could have. All right, so let's take a look at some examples. All right, so I have two pedigrees here. One's going to be representing a dominant trait, and one's going to be representing a recessive trait. So when you are looking at a pedigree, you can look at some patterns, and it might seem kind of confusing at the beginning, but I'm going to try to encourage you to look for a particular pattern that's going to help you to determine whether it is dominant or recessive. So the particular pattern you want to take a look at is see if there's any kids in any of the generations that have a different phenotype than the parents. Okay, so let's look at this one first. So in this first box on the left, in this first pedigree on the left, look at any of the matings of the parents. We have 3 generations in each of these, in this family tree, so to speak. So we have a couple sets of grandparents and some kids and then some grandkids. So look for any combination of parents and kids where the kids don't have the same phenotype. The phenotype is what you see. We don't know the genotypes yet. We haven't figured that out. So shaded versus unshaded. So if we take a look at this first family, we can see here those kids and the parents have all of the same phenotype. That's not going to help us out. If we take a look at this family, we can see that the mom is unshaded and the dad is shaded, and they have 2 kids, both shaded and unshaded. So there is a representation of the offspring of each of the parents. Now, if we take a look at this family right here, we do see an offspring, this one right here, that is unshaded, that is different than both of the shaded parents. So that is your clue. Okay, so that's the family we're going to look at. That's the family that's going to help us figure out this situation, whether it's dominant or recessive. And so now what you can do is set up some Punnett squares to do it like a trial and error. Okay, so let's just guess. Let's say that it's a recessive trait. So if they're affected by this trait, recessive, say albinism, is it a recessive trait? So for you to be albino, to be affected, you would have to have little a. That's the only way that you could do that. So little a would be like a shaded box. Okay, you're affected. So what does that tell you about the unaffected genotypes? They would have to be big A, or big A, little a. So these would be, I'm just doing a square, but it could be square or circle. All right. So if this pedigree is of an individual or a family who has a recessive trait, we'll just say albinism, but we don't know yet. Let's go ahead and put in those particular genotypes and see if we could get the outcome. So if our shaded boxes are AA, we would have an AA here and AA here. So if both of those parents were albino, they would have albino kids. Oh, but look at individual number 7. Individual number 7, if we go down to our key, has to be either big A or big A, little a. They have normal skin pigmentation. Is that possible? Is that family possible if this trait is a recessive trait? What do you think? Maybe by seeing that I'm hesitating, the answer is no, that's not possible. If this was a pedigree of a recessive trait, there is no way that 2 parents that are homozygous recessive could have any offspring that have a dominant allele. It is impossible. So that lets us know by just this trial and error attempt that this is not a recessive trait. And it is actually, let's see if my animations are working. It is a dominant trait. Here's that polydactyly. I had the picture earlier with our dominant recessive slide. This is a trait of polydactyly. It's showing a pedigree of people that have 6 or more fingers on their hands or toes on their feet. So if we were to set up our key, then that means kind of what we have here. These are going to be shaded in boxes. Our dominant, homozygous dominant, or heterozygous are going to be the affected individuals, where unaffected individuals are going to be homozygous recessive. So if we're to go and start filling in that known information, we can start attaching these genotypes to these phenotypes, which are either the affected or the unaffected. So I think I have this with my little, there we go. So if you have 5 fingers in this particular pedigree, you have to be homozygous recessive. That's the only genotype that you could have. Okay, so those are easy to fill in when we know that it's the homozygous recessive because that's the only choice. Now, if we take a look at individuals 1 and 2, they could either be homozygous dominant, the big P, or homozygous or heterozygous, big P, little p. Now, if they had a kid, individual number 7, that was little p, both parents had to have been able to donate a recessive allele. That's the only way number 7 person, man, boy, number 7, could have gotten homozygous recessive. So what does that tell you the genotypes of individuals 1 and 2 have to be? They are 6-fingered, 6 or more fingered, so they have to have a dominant allele, but they also have to have a recessive allele to donate to their son. So that tells you the individuals 1 and 2 have to be heterozygous for them to be able to be 6-fingered and have a 5-fingered kiddo. Now we can go and fill in the rest of their family, and we may not know exactly what phenotype or genotype they have. Oh, sorry, we're jumping over to family number 2. So same idea with family number 2, the individuals 3 and 4. So we know 3 is 5-fingered, 4 is 6-fingered. So we know that 4 has to be heterozygous because that's the only way they could get individual number 8. Okay? So now we can go in and fill in these other individuals. So how come in this one family on the left, 5 and 6, they could be either one? Well, you could do a Punnett square. When you have heterozygotes mixing, you could have any one of those combinations. So when you have individual number 1 and individual number 2 mating, you could get an individual, that 3-to-1 ratio of the dominant phenotype could be either heterozygous or homozygous dominant. If we take a look at individual number 9, it can't be one or the other because mom had to have donated a lowercase p, dad had to have donated the uppercase p, the dominant allele, for that individual to have the 6 fingers. All right, so that is an example of a dominant pedigree. Now let's take a look and move over to this other family. So again, like I said at the beginning of the last one, look for a family of some parents and some kiddos where a phenotype of a kid does not match the phenotype of the adult. Okay, so let's look at this first family. Well, we've got one of each in the parents and we have representation of each in the kiddos. So that's not one to work with. In this family here, it's the same, mom and dad are one of each. And we've got a smattering of that in the offspring. But if we take a look at this family here, take a look at what we have. We have 2 parents who are unaffected that have a daughter that is affected. So just like we did before, we can do a trial and error. Okay? So let's set up a Punnett square for that family. And let's say maybe it's another dominant allele or a dominant trait. So dominant means they would have it because that's the trait you're following. So if the shape is empty, then they don't have it. So that means they would have to be little a. So if we did a chart, we would see that 100% of the offspring would have to be homozygous recessive because they are unaffected. But we have individual number 4 in that second generation that has a dominant allele. So there's no way that these unaffected individuals are going to be homozygous recessive. which was our dominant, the shaded boxes we were going to say was our dominant allele, right? So this actually is an example of a recessive trait. So where the recessive trait is going to be our homozygous recessive individual. The shaded shapes are going to be our homozygous recessive. So this is actually a pedigree of albinism, where big A, and big A, little a, these would be our shaded in boxes. Right? Normal skin pigment, where little a, homozygous recessive would be our empty boxes. So again, no, that's backwards. Sorry. Let's correct that. Normal would be empty, and the albino would be a shaded in box, because they're the ones that's affected. They're the ones that are showing that trait that you're tracking in a pedigree. So if we were to go ahead and assign the individuals who are homozygous recessive, again, that's the easiest part to start. They are going to be the shaded shapes. So we can fill those in pretty easily. Okay, so now we can go and start filling in the rest of the family. So in this first family here, this one, we'll highlight right there, if mom is albino and they have an albino daughter, dad has to be heterozygous, because if he was homozygous dominant, 100% of the kids would be heterozygous. There would be no chance to have a homozygous recessive offspring. So dad has to be heterozygous. And in that case, the kiddos are also going to be heterozygous, because the mom will always donate the recessive allele. And if they're not albino, that means the dad donated the dominant allele. In this other family here, in this family that allowed us to determine whether it was dominant or recessive, if you have two non-albino individuals, they're showing the dominant phenotype, but they had an albino offspring, which means they had to have both donated a recessive allele, which means they have to be heterozygous. So individual number 4 in that second generation is an albino, but the brother is not. So that means the brother has to have at least one dominant allele. So in this case, it could be either homozygous dominant or heterozygous. Now in this second the grandkid generation here, in this family, we've already determined the parents. We have a heterozygous dad and a homozygous recessive albino mom. They have a couple albino children. What are going to be the genotypes of individuals 2 and 4 in that third generation? Well, they have to be heterozygous. There's no way they can be homozygous dominant, because mom was only able to donate the recessive alleles. All right, so it's kind of like solving a puzzle. It's like a mystery. So each one of these matings, you could draw out a Punnett square, and you would have the potential, the chances, or the possibilities of the offspring. But then the information, the pedigree chart, tells you what the actual outcome of that particular cross was. And then you can go back multiple generations to track where that trait may have come into the family and how it tracked through generations of that family. All right, kind of a cool genetics tool. All right, that's it for pedigrees. I will see you next time.